Okay, so osmoregulation is the process by which kidneys will remove the waste and selectively reabsorb useful substances back into the bloodstream. So osmoregulations take place in the nephrons, which are basically long tubules surrounded by capillaries in our kidneys. Structure of the nephron consists out of renal capsule with glomerulus, proximal convoluted tubule, or in short PCT, loop of Henle, distal convoluted tubule, and collecting ducts. So basically here we have four main stages of osmoregulations that will take place during our blood filtration. The first step, ultrafiltration, will be happening in the glomerulus and the renal capsule. It will be a process which will be based on forcing small molecules through some filters, which will be discussed later on, against the concentration gradient into the filtrate. Next, we will be looking at the selective reabsorption. This is the process by which in the endothelial cells of proximal convoluted tubule, all of the glucose will be reabsorbed back into the blood for the sake of the conservation. Loop of Henle and DCT and collecting duct processes will be basically based on the osmotic gradient. We will look how water will be reabsorbed back into the blood by diluting the solution surrounding the basically capillaries. So yeah, that's basically it, and we will go to the first step. Okay, so before we talk about the filtration process itself, we need to talk about the filters themselves. So basically, the first layer would be our capillary walls, our endothelial cells. Next we will have gel-like basement membrane, and next we will have podocytes, which are basically will be epithelial cells of our nephron. So the main things that we should filter out of the blood will be urea, glucose, some ions, and amino acids with water. The things that shouldn't be filtered out, that we shouldn't really find in the filtrate, will be things like big red blood cells or blood cells in general, and big polar proteins. That's because they wouldn't be able in a normal body to pass through those gaps in the epithelial cells of the podocytes and endothelial cells of capillary. So in our basically nephrons, we will have blood coming from the afferent arteriole, which is coming from aorta, which on its road will basically decrease in diameter and form efferent arteriole. This will basically cause an high increase in hydrostatic pressure, which will force out small molecules through those type of filters, such as those gaps that we talked about. And the filter would basically pass and form a filtrate in the Bowman's capsule, this yellow stuff you see on the basically diagram. Yeah, and that's basically it for the ultrafiltration. So we are left with everything in the filtrate except for big proteins and red blood cells. In the next step you will see how all the glucose is getting reabsorbed. Okay, so here is our new diagram, treated as the continuation of the previous one. So basically from our Bowman's capsule we come into the proximal convoluted tubule, or in short PCT. So we have our cilia and endothelial lining on PCT in green, basement membrane and capillaries, which are basically surrounding whole nephron in order to reabsorb all of the stuff that may revolve around the nephron itself. Okay, so now let's talk about how all the glucose should be reabsorbed in the normal kidneys, and we will begin from mitochondria producing ATP. This ATP would be needed to supply energy in order to pump sodium ions from the endothelial lining of the PCT to the blood directly, basically through the basal membrane and all of that. So look, we are pumping with the concentration gradient, so the concentration inside of the cell would be really, really low. This means that quite a bit of a sodium will be diffusing from the filtrate into the basically cytoplasm of our endothelial lining, but this time it will travel through co-transporter, which is basically a passive movement, but this passive movement of sodium with their concentration gradient will also supply energy to move the glucose with itself, so basically we will also transport glucose with our sodium passively. We don't really need any energy for that. This will cause our concentration of glucose to rise in our endothelial lining cytoplasm, and because we have quite high concentration of glucose in the filtrate and quite low in the blood, our glucose in the cell will basically choose the pathway of least concentration to fulfill basically the equilibrium and will travel along the blood. That's how all the glucose in the body is reabsorbed in the basically healthy patient, healthy person. Okay, so now we will discuss the loop of Henle step. In this step, basically we will reabsorb as much of water as possible back into the blood. In order to do that, we have basically a descending limb and ascending limb in our nephron. Note that they are placed in the medulla and that the ascending limb has the thicker wall. Now we will discuss why would we really need those descending and ascending limbs. So descending limb is the one which is permeable for water as impermeable for solids. So it will basically allow our water to leave the nephron. And the ascending limb has those thicker walls, and those thicker walls have basically a lot of mitochondria inside of them. And this ATP will be used in order to basically pump the sodium ions that were left from the filtrate to the intermedullary space. This will cause the concentration of solid in the intermedullary space to increase and will cause basically water to diffuse with the concentration gradient because of this osmotic gradient formed by the sodium ions. Yeah, and here our arterioles basically wrap around our loop of Henle, which allows our water to be reabsorbed. Yeah, and that's basically it, and now we will move on to the distal convoluted tubule and the collecting duct. You will see that it is also about water being reabsorbed, however the different method is fairly used. So yeah, here is our new diagram. Here we see our distal convoluted tubule and our collecting duct on the right. Before we go into the basically mechanism of how the water is reabsorbed, we need to understand that hypothalamus has osmoreceptors in it. Those osmoreceptors allow our body to identify if our blood is dehydrated or overhydrated. If, for example, our osmoreceptor detected that our water content of the blood is too low, it will send action potentials to the hypothalamus, which in turn will basically cause its pituitary gland to release hormone that is called ADH, antidiuretic hormone. This hormone will basically travel via blood to our distal convoluted tubule and collecting duct and will basically bind onto the first messenger receptors on the epithelial cells of those DCT and collecting duct, and this will cause a certain cascade to be started in those cells with the receptors, which will cause the cell to transcribe and translate more genes and basically mRNAs that code for aquaporins, which are basically a sort of a protein channel which will allow you to release water. And now you may ask a question, why should it really leave? So basically that's the same thing as in the loop of Henle thing. We have fairly a very high concentration of solid outside and quite low inside comparatively, and that will basically cause the osmotic gradient and our water molecules will leave and basically will be reabsorbed back into the blood. That's it. Here is also the structure of basically a kidneys for those who are interested. You don't really need to know all of those structures. The only thing that you need to know is basically the structure of the nephron itself. And the only two parts that you also need to know is basically the existence of medulla and the cortex, and that the arteriole that is going into the nephron is afferent and the exiting arteriole is efferent. And that's basically it. Rest of it is basically just for your own knowledge.